Welcome to Microfluidics Unleashed, where we explore how lab-on-a-chip technology is reshaping diagnostics and research. In this presentation, we'll walk through manufacturing, pumping, valves and mixers, droplet systems, digital PCR, and real-world applications like single-cell analysis and wearable sensors. Think of this as a guided tour of how tiny channels and smart design enable big advances in biology and medicine. This slide compares common manufacturing paths, from soft lithography and PDMS prototyping to thermoplastic injection molding and femtosecond laser machining for advanced 3D structures. Rapid PDMS prototyping is fast and low cost, but has absorption limits, while thermoplastics like COC and PMMA scale well for mass production with good optical clarity. Laser machining offers high resolution for complex networks when scalability is required. Here, we contrast syringe pumps and electro-osmotic pumps as core ways to move fluids on chip. Syringe pumps deliver very stable, long-term volumetric flow ideal for cell culture and extended assays, whereas electro-osmotic pumps enable fast, integrated switching without moving parts, great for rapid response and dense on-chip routing. The illustration highlights steady flow versus rapid, switchable control for different experiments. This slide breaks down two key microfluidic components: Quake-style membrane valves and Tesla mixers. Quake valves use pneumatic deflection of a PDMS membrane for millisecond channel closure, enabling complex routing and rapid switching, while Tesla mixers exploit chaotic advection at low Reynolds numbers to drive complete mixing far faster than straight channels, which is crucial for efficient reagent blending in compact systems. Droplet generation relies on simple geometries like T-junctions and flow focusing to create uniform, monodispersed droplets for high-throughput assays. The images show how shear and pinching produce consistent droplets. Then surfactants stabilize them to prevent coalescence. This approach yields picoliter reactors for single-cell or single-molecule assays, enabling massive parallelization and precise quantitative readouts across thousands of compartments. Digital PCR turns bulk DNA into thousands of discrete droplet partitions, allowing absolute quantification of target molecules without standard curves. Positive droplets follow Poisson statistics, so counting positives gives precise, single-copy sensitivity. The slide also highlights transformative applications, like liquid biopsy for ultra-rare mutation detection and sensitive pathogen surveillance, where absolute counts enable confident clinical decisions. This page shows single-cell workflows enabled by microfluidics, from hydrodynamic trapping arrays for time-lapse imaging and secreted protein profiling, to microfluidic sorters that use dielectrophoresis or acoustic waves for precise separation. These tools support transcriptomic barcoding and high-purity isolation of rare circulating tumor cells, delivering the sensitivity and throughput needed for modern single-cell biology and diagnostics. Organ-on-chip designs recreate tissue-tissue interfaces by layering an epithelial channel over an endothelial channel, separated by a porous membrane. The illustrations show lung alveoli with air-liquid interfaces, liver sinusoids for drug metabolism and hepatocyte interactions, and kidney nephrons for filtration and reabsorption, demonstrating how dynamic microfluidic environments yield more physiologically relevant responses than static well plates. Paper microfluidics uses wax printing to define hydrophobic barriers and capillary channels in nitrocellulose, enabling pump-free flow for cheap diagnostics. The colorimetric assays illustrated detect glucose, proteins, and nucleic acids in a multiplexed format, with results visible by eye or phone. These devices cost less than $1, making them ideal for resource-limited settings and at-home testing where accessibility matters most. This slide highlights mobile and cloud-enabled diagnostics for real-time surveillance, showing phone-attachable readers that quantify fluorescence or absorbance, and upload GPS-tagged results to the cloud. Secure transmission and analytics enable epidemiological mapping and environmental contaminant tracking, creating a feedback loop where field data informs public health responses and supports rapid, decentralized monitoring of infectious diseases. AI-driven optimization accelerates microfluidic design by tuning channel geometries for better mixing, particle focusing, and droplet generation in minutes rather than weeks, as illustrated here. Paired with generative design for 3D architectures, these tools produce complex, high-performance structures that are hard to imagine by intuition, enabling rapid prototyping, targeted solutions for rare samples, and automated customization for new applications. The flexible sweat sensor slide shows soft lithography on elastomeric substrates to create conformal microchannels and integrated electrochemical electrodes for continuous metabolic monitoring. Sweat is routed through microchannels to detect lactate, chloride, and cytokines, with on-chip electrochemical readout enabling real-time insights. These devices open non-invasive pathways for personalized sports medicine, health tracking, and optimized performance monitoring.